I think it's not, we cannot say it's good morning, good evening, or good afternoon. So I welcome all of you, and I will open the floor to Floor, who will start the seminar series. Floor, take on.
Thank you, Abraham. Well, good morning, good afternoon, or good evening to everyone joining us from around the world. We are truly excited to welcome you all to this first online event of this yICSA webinar series. It's fantastic to see such a global community with over 100 registration from different corners of the world. And we hope this will be a motivating initiative. We are especially grateful to our speaker, Eleanor Sheekey, for being here with us today. So thanks, Eleanor. So without further ado, I will now hand things over to Nichelle, who will explain how this first online event will unfold. So Nichelle.
Thanks, Flor. And thank you all so much for your support by registering for this event and joining us. We think it's going to be a really exciting series. One of the things that the yICSA community has noticed is that we have a very vibrant senescence field, and there's so much exciting research that's going on. And so the yICSA webinar series really aims to provide an online platform for us to connect regularly and share that science, and hopefully create engaging scientific discussions between PhD students, early career researchers, and PIs. So the main format for the webinar will involve a talk, which is by our invited speaker, and that will be followed by a moderated Q&A session. We're absolutely thrilled that Eleanor has accepted our invitation to speak today, and that will be followed by a Q&A session that is moderated by Samir from the yICSA committee. So before we get into this very exciting webinar, I would like you to join me in inviting Han Lee from the yICSA committee to say a few words and officially inaugurate the yICSA webinar series. Han, over to you.
I think you can hear me? Okay, all right. So it's really a great honor for me to join you today as we officially kick off this exciting webinar series organized by the wonderful and dedicated yICSA team. This initiative reflects the growing momentum in the senescence research and demonstrates our collective commitments to advancing our understanding of this critical field on a global scale. So the yICSA is an open, inclusive, vibrant, and international community with many young researchers. This webinar series is a wonderful reflection of that spirit. So of course, this platform is designed not only for young career researchers to share their ideas and experience, but also for everyone in the community to engage, collaborate, and spread the knowledge. So I encourage all of you to help us in spreading the word about the upcoming webinars and fostering a stronger network of scientists working in the senescence field in the global scale. Thank you all for being here, and we look forward for the stimulating discussions in the webinars ahead and the organic collaboration that will follow. Thank you all.
Okay. Thank you very much for the inauguration and the introductions. My name is Samir and I'll be chairing today's session, so welcome to everyone. It's my absolute pleasure to introduce Dr. Eleanor Sheekey, who carried out her BA and MSCI in biochemistry at the University of Cambridge. It was during her undergrad where she started a Sheekey Science blog that eventually turned into a YouTube channel, which started in 2019, that I'm sure we'll hear about later on. Eleanor completed her PhD at the Cancer Research UK Cambridge Institute with Professor Masashi Narita, where she studied cellular senescence and the role of p53 mutations in response to DNA damage. She's since moved to Japan, I think 3 months ago, to start a postdoc with Dr. Fujiwara, investigating the skin extracellular matrix and aging. So today her talk is entitled Stop, Pause and Play, From cellular senescence to YouTube. Eleanor, the floor is yours.
Thank you. Let me just share my screen. So I really hope that you're looking at the right slides and not my presentation modes, and you can hear me. Okay, awesome. Thank you so much for inviting me. Firstly, it feels quite a privilege to be the first speaker of this webinar series, which is therefore somewhat terrifying, but also excellent because I get to set the stage for how everyone else has to sort of proceed after me. So that gives me some freedom to talk about what I want a little bit today. And so, as Samir stated, I'll be talking to you today about some of my PhD research in cellular senescence, which is perhaps where the stop part of my title came from. And then we'll have a bit of a pause where I somewhat reflect about where I currently feel the state of the senescence field is, and I guess my own personal academic reflections. And then also the play aspect refers to the science communication that I've been doing, which even before the YouTube channel I was doing during my undergrad. So hopefully this will be somewhat lighthearted, also because it's being recorded. Unfortunately, there won't be as much detail about my exact research as maybe I would like to be able to share, but I have presented some of it before in different conferences, and obviously offline I'm happy to speak with people about it. But yeah, hopefully this will be enjoyable and still exciting nonetheless. So exactly what I'm trying to set out today is to take you from senescence and connect you through to YouTube. So I think I've already said exactly the outline. Unfortunately, my title was a bit backwards and I probably should have started with play, but I think it makes most sense to start with my academic research. But as I also realized that some of you have no idea who I am, and I also thought I would talk a bit about myself. I am British, and I actually, before wanting to be a scientist, I wanted to be a magician when I was younger. I have since realized that being a scientist also is somewhat like being a magician to some extent, perhaps. But I love learning. I think that's something that I've always had growing up, and obviously is reflected in an academic career and also science communication. But I also have a life outside of the lab. I quite active. I do like running, playing football, and watching movies. So that's the sort of fun part. My academic career, as Samir correctly stated, I did my undergraduate in biochemistry at the University of Cambridge. This also included an undergraduate master's degree, which I did with Professor UC Taperley's lab, studying CRISPR-Cas9. After that, I then joined Masashi's lab at the Cancer Research UK Cambridge Institute, which is mainly the research I'll tell you about today. During that project, I was very lucky to have the chance to spend a summer in Japan, in Osaka. And I think, as I'll probably mention later on, this is one of the reasons why I then decided to come back to do a postdoc here in Japan. And then I actually finished my PhD December last year. And so before coming here, which was in the end of June, I actually went back to the biochemistry department for 6 months to work with Marc Delaroche. I won't really talk about the work I did with him today, but there is still a senescence link, I can assure you. And then now, as of the 1st of July, I joined Hironobu Fujiwara's lab, and I'll briefly mention what I'm hoping to do in that lab. But alongside that, I've also always had a strong science communication passion, and that sort of begun during university, where I would write blogs for this website, the Journal of Young Investigators. And then even before starting YouTube, which was in 2019, I had a blog that I used to write, and then I translated that to YouTube. But before we get to the play part, let's actually talk about my interest in senescence. And so I suppose the general gist about what I was interested in my PhD was how does a cell respond to stress and enter the state that we all are interested in, which is cellular senescence. Now, I wasn't entirely sure how much detail I should give about senescence, given you are all experts as much as I am, but obviously I know that this is to spread the knowledge to a wider audience as well. So my sort of biased opinion is considering the role of p53 in the senescence induction process, and how I view senescence is as a cell fate that happens with key characteristics, of which the two that I think I was most interested in my PhD was this exiting from the cell cycle and this irreversible cell cycle arrest, and the development of the senescence-associated secretory phenotype. So while I've brought up p53, it's important to reflect that p53 isn't necessarily just involved in senescence, but has many functions within a cell. And following a stress event where p53 gets activated, it might be able to trigger downstream responses that help to resolve the stress such that the cell can return back to normal, or it could enter into senescence, or it could induce alternative cell outcomes such as apoptosis. And so really, I like to think of p53 as a sort of decision maker. However, at the moment, it's still unclear exactly how this decision is being mediated, and if it is just p53, or what exactly the regulatory steps are involved in these different outcomes. So when it comes to considering p53, firstly, what exactly is it? Well, p53 is a transcription factor, and so it can bind to DNA and regulate the expression of different genes. And due to the fact that it gets activated in response to stress, it perhaps is not surprising that it's one of the most commonly mutated genes seen in human cancers. And so when it comes to understanding the role of a protein or a gene, one thing that you can often do is see what happens when you have a mutation or a perturbation in that gene or protein. And so as much as I was interested in looking at the consequences of mutations in p53 from a cancer perspective, it also gave me the opportunity to see how the usual pathway gets perturbed in response to these mutations. And so, in other words, there are many different ways in which p53 can get mutated, and I decided to just pick one main mutation that I focused on that commonly arises in human cancers, and this is a so-called missense mutation in the DNA binding domain, where you go from arginine to histidine. And so this missense mutation means that the full-length protein still gets produced, it's just in a single amino acid substitution. And my project was really trying to consider how this mutation could impact the response to cell stress. So as I've already stated, p53, in terms of its wild-type function, is thought to act as a tetramer when it interacts with DNA to activate different genes. However, I was interested in what happens when you have both wild-type p53 and mutant p53. And so there are various different outcomes that could happen when you have a mutation. Firstly, the mutated form, because it's in the DNA binding domain, could perturb its ability to actually interact with the DNA sequence, and therefore has a simple loss of function outcome. So if we take a look at a gene that's commonly regulated by p53, we would expect loss of function to have a 50% reduction in a heterozygous state. Now, whilst it would be nice if it was that straightforward, there are two other possibilities that might arise when we have both wild-type and mutant present in the cell. One of which is dominant negative activity, which arises due to the fact that this tetramers could form that are so-called heterotetramers, whereby you have both wild-type and mutants present at the same time, with the mutants effectively taking away the wild-type function. So therefore, it could be even worse an impact on gene expression than just the simple loss of function. And then the last outcome could be gain of function, where the mutant protein is doing something completely novel that isn't seen with the wild type protein. And so this could be activating different genes by indirectly interacting with the DNA sequence with other transcription factors. So my thesis was quite big in the end, and also I haven't got time to really go into all of the details today, unfortunately, but this is the general overview about I was trying to address in my project. What happens when we have mutations in p53? And how does that impact the stress response? And does that tell us anything about tumorigenesis or ways in which we can understand aging within tissues, as these mutations in p53 also accumulate in different tissues with age? And so ultimately what I needed to do was to have a good model system to be able to investigate this question and how to go about distinguishing between dominant negative versus gain of function effects. So what I decided to do to distinguish between these two different functionalities was to actually create two different cell lines, one of which knocked down wild-type p53 to mimic a sort of loss of function impact, and another one was to overexpress the mutant protein. And therefore, by comparing these two different cells at the same time in response to stress, changes that happened both with knockdown or with overexpression of mutants would reflect things that are dominant negative activity, and genes that were upregulated in the presence of mutants p53 but had no impact or little impact when p53 wild type was knocked down would more likely reflect gain of function activity. Now, this is probably a little bit complicated to understand and explain on a Friday evening for myself, so apologies in advance. But these were the two different, these are the constructs that I was using to overexpress mutant p53 in my cells. And I was working with normal human lung fibroblasts. So essentially, I was trying to mimic a heterozygous state whereby I had both wild-type p53 and overexpression of mutants. And then the fun part was then inducing senescence by inducing DNA damage through the treatment with etoposide. Now, to cut a very long and hard story short, one of the most interesting findings from my study was simply by just comparing the gene expression profiles from these two different cell lines in response to stress. And I think what's most striking is, especially in the acute DNA damage response, so this is 24 hours in response to etoposide treatment, there's a very strong correlation between the two cell lines. So cells that have high expression of mutant p53 and cells that have knockdown of wild-type p53. And so this really showed that a lot of the activity was simply a reflection of this dominant negative activity. And you can observe that in senescence, that this correlation is still present, albeit a bit less. And this might actually just reflect the activity levels of p53 in these two different states. So acute versus chronic stress levels. Now, I was also interested in investigating this gain of function. And in particular, I was curious as to why I could observe some gain of function genes potentially in the acute phase, not in senescence. And so when I took a closer look at these genes, what was most interesting was a lot of the genes that were gain of function in the acute stress, but not senescence, were senescence-associated genes, in particular, a lot of inflammatory factors. And so the reason that these gain of function genes get lost in senescence is because these genes are already being upregulated as part of the secretory phenotype. So in other words, the gain of function signature becomes masked during senescence. However, that said, there were a few genes that I could classify as persisting as being more highly expressed in the mutant state compared to the control cells. And so this is when I should also have remembered to tell you that high is referring to high expression of mutant p53. But I think this is one of the main findings from my research in general, was this interesting change in the gain of function signature. In other words, that it's very much context dependent. And so this obviously raised many, many questions, one of which was, how exactly is this gain of function being mediated? And this is something that I've had to take out for today's webinar, and instead you will have to put up with some pixelated pictures of potential possibilities as to what might be happening. I did investigate this in my PhD, but just for time reasons today, I won't talk about it. But instead, I wanted to talk a little bit more about the changes in the secretory phenotype, as a lot of these gain of function genes were linked with inflammation, and to consider if there was any functional consequences. So to do that, I decided to do some mass spectrometry of the SASP. So by having the standard DNA damage-induced senescence model, and then what I refer to as MSASP, the mutant p53 secretory phenotype, and then compared them in senescence and control, and then between wild type and then heterozygous mutant cells. And because proteomics data is never as nice as RNA sequencing data, to have a more sort of solid understanding about these changes, I decided to compare the RNA sequencing changes with the proteomic changes. And so, again, just for matters of time, all I really want to say is that there were some consistent correlations between genes that were upregulated from the RNA sequencing data in my mutant p53 cells that were also seen as being more highly expressed in the secretory phenotype. And this gave us some interesting insight to say that the SASP does appear to be different in these heterozygous mutant p53 cells. So given that the secretory phenotype we know is involved in secreting things like cytokines, inflammatory factors, matrix metalloproteases, it suggested that maybe these cells have an impact, a functional impact in the organism if they arise, and therefore could have functional consequences. So most of my research was in vitro, but naturally I'm more curious about how does this happen in vivo, in physiological terms, when we have these heterozygous cells arise due to random mutations. If there's some stress that occurs, do the cells expand and outcompete the neighboring cells, or do they get selectively eliminated because they're having this sort of premature senescence-like secretory phenotype? And so this is why I ended up in Japan last summer, as I tried to investigate this in zebrafish as a model organism. And again, I unfortunately haven't got time to talk about it today. But essentially, this was this concept I was trying to investigate, which is this concept of cell competition, how cells, given the same stimuli, have different responses, and how that can impact the downstream outcome. So I'm sorry that was a complete whiz through my PhD, but there was a lot to talk about. But hopefully at some point my research will be published and you can read about it in more detail. But for now, I thought we would take a little pause to just sort of reflect and digest some of the stuff that I just sort of told you all in rapid detail. And actually, I think that there is only one real take home I want to give from this whole presentation, and I've already subtly put it into my presentation. And I think that the key message that I want everyone to remember today is this importance of communication. Now, you may have noticed that my slides were a bit chaotically organized, but that's partly because I was trying to hide within my slides the word communication, as I think it really is one of the most important sort of connecting features between both my academic and research interests, as well as my science communication, and also now my personal situation living in a country where I struggle to communicate day to day because I am, as of yet, not fluent in Japanese. So that then brings me on to some of the science communication work that I've been doing, and my interest in doing science communication on YouTube. So as I mentioned, before actually starting on YouTube, I actually had a blog page, which I don't recommend you checking out, as if you can read any of this, you won't find my innermost secrets, so it's therefore not worth looking at it. But for me, I think it was just a bit of fun, and for me, at least, when I write things down, even if it's not for other people to see, it helps me to process my own thoughts and logic over complicated topics. So that really was the origin of my YouTube channel, was kind of a more selfish me trying to sort of just process information that I read and was trying to understand. And naturally, I was never the best with words, as you might be able to tell, but I liked drawing because I was definitely a very visual person. And so after that, I eventually then found the way to sort of talk about research papers in a video format. And so half of my videos, well, about half, where I try to digest different topics or research papers that I find interesting. And then the other section of my YouTube channel is where I attempt to use my bad British humor and make researchers laugh. In other words, I have the privilege to get to interview many researchers around the world and to talk about their research, and ultimately try to use it as a platform to showcase some of the really cool work that people are doing, which is also why I'm very honored to be doing this with the yICSA community today. As one of the most rewarding aspects from doing YouTube has really been the ability to meet other people and to foster networking and a collaborative environment. And it's led to me being able to both present and host at conferences, and even just going to schools has been just like a really good thing to do, as one of the things I still want to do like for the rest of my life is just to try and be a good role model. And so being able to excite people about science, for me, it's like one of the most stimulating things that I can do. Now, the challenge is, how do I balance doing something like YouTube with being a researcher? And well, fortunately, there is some overlap between what I talk about on YouTube and my research, albeit this would be the the hopeful situation where everything that I'm wanting to understand falls within my YouTube content. However, it's a little bit more chaotic than that, because I think that the more I sort of read outside of my topic, the more my research field expands, and then that expands my reading outside. It's just a bit more like this, in my opinion. So there is overlap, of course, and I think you can always go down different rabbit holes in science. And I think that, if anything, trying to read papers that are very much not related to your complete focus can often give you creative ideas and find connections between two different topics that might down the line help you out with your own research. I'm not sure if that made sense. It made sense in my head, but I think that there is something to be said about trying to read more broadly as opposed to being fixated on your specialty. In other words, just check out some of my videos. Actually, yeah, speaking of, one of the most recent videos I made was on this concept of tissue rejuvenation and a process called polydenosis, and this is with Professor Jason Mills. And this caught my interest because, well, my favorite protein was there, but for other reasons as well, that it's a process stimulated in response to injury. And I highly recommend this review article if you want to learn more about the polygenosis process. But essentially, it was just raising different ideas in my head about how it might change our understanding about how cells respond to stress, and how I mentioned earlier that P53 can induce things like senescence, could induce apoptosis, but it's also now involved in this paleogenesis pathway, this regulation of the re-entry into cell cycle. And so I just feel like the more I learn, the more confused I get with biology, but I think that's somewhat natural as well. And so this lead me on to talk about what am I actually working on now, and what do I think more broadly we should be focusing on in the field. So I'm now living in Japan, is the first bit of news that you already know. In other words, I had to have a Japan to get me here in the first place. And so after coming last summer, as part of my PhD research, I knew I wanted to come back. And so that obviously involved trying to find a job. But more broadly, I had to also think deeply if this was actually a place I wanted to come back to. And so I did look for jobs elsewhere and considered also both academic and non-academic positions. As with any job application, there's interview processes. And fortunately, I was already learning a bit of Japanese, which was helpful. Then I wanted to find a lab where I could do good science. I think that there are so many amazing labs out there, and I think that didn't make the decision process any easier. But ultimately, I wanted to go to a lab where I could learn some new methods, meet new people, and learn a language. And then for me as well, moving countries also meant saying goodbye to a lot of friends and family that were obviously in close proximity. So I wanted to also make sure I had a good life outside of the lab. And obviously I've only been here 3 months, and so far so good. But for me, it takes a lot of proactive effort to try and reach out to people and to sort of have a good work-life balance. But besides doing good science and having fun, my mission is to also try and spread senescence, obviously not just in Japan, but globally. And I realized that this kind of looks bad because we talk about spreading senescence spread as being a bad thing. But in this case, I think it's a good thing. I just need to work out how I can make it in a good way. And so without giving away too much about what I'm hoping to do, I'm now in a lab where there's good resources to trying to understand the extracellular matrix, which I think understanding senescence and changes in the ECM is something that we have a growing understanding of, but there are still so many interesting questions, not just in senescence, but also just more generally in tissue aging. So that's something that I'm hoping to focus on. And for me as well, the novelty is that I'm for the first time doing mouse work, which is both challenging but also interesting. And I guess leads me on to some of the more general questions that I think we should be thinking about in the space. It looks a little bit chaotic. I think, yeah, there's always endless questions that you could have. But I think I'm still curious about how exactly the decision-making process is happening in the cells. What exactly are they responding to when we say injury or stress? What exactly does that mean? And then from that, considering the response of senescence in different tissues, how does that change if that cell is mitotic, post-mitotic, and really questioning what really distinguishes a senescent cell from an aged cell, particularly if that senescent cell came from a post-mitotic cell. And I didn't want to have to read all of these different bullet points, but I think, yeah, there's a lot of, I mean, I know as well, there's many labs doing research on these topics. But I think, yeah, I'm excited for hearing about other people's work. And I think that this is really the best time to be in the senescent space. So I think we really can have an action on some of these questions. And I realize I've presented it in a sort of chaotic, crazy manner. So I apologize for that. But I really think that this is the chance for me to also say that we as a community are working together, not against each other. And as well as being on the same team, I always like to say that you can't compete with someone who is having fun. And I think that for me, challenging questions are also the most fun questions, because they're always so stimulating. And so just to sort of wrap up my talk, I thought I would try and give some unsolicited advice and goals that might be helpful. I think I probably mentioned most of these already. I think reading broadly in space and time, I mean both in terms of topics, but also time in terms of how old was that literature. I think a lot of some interesting ideas I've had most recently have come from reading maybe 50-year-old papers or books. So there's always creative ways to come up with ideas. I like going for very long walks. I don't know if they're ever helpful or just for me entertaining, but that helps me. I think it's important to remind yourself why you're doing what you're doing. What is it that drives you as a scientist? Is it curiosity? Is it to find a therapeutic application? Or maybe you don't know why you're doing what you're doing, but maybe you should think about it. I don't know. I think obviously you should take science seriously. And I like to think I do take science seriously, but I also do think it's important to have fun. And one of the last things that I've come to notice I've always known this, but has been very evident living in a country where for the first time I've had to really struggle to communicate day to day, that you might not be the best at everything, but there's one thing that we can all be good at, which is kindness and showing kindness to other people. And I know that this community already is a very kind community, so I didn't really need to say that. But yeah, I think that's almost everything, because this slide always cracks me up, because I think trying is also one of the most important things that you can do. This was the first sketch of my YouTube character. Fortunately, it got better. And then lastly, whenever I feel sad, I like to remind myself that at one point I was just a single cell. So it's just incredible to know that we even exist. So with that, thank you for listening, and let's keep this community going, and keep communicating ultimately. And yeah, come visit me if you want to. I hope to see some of you again in the future. So yeah, thank you again for letting me be the first speaker. I hope I've maintained relatively good standards. And yeah, happy to take any questions.
Thank you so much, Eleanor. That was really great, and I'm sure this is mirrored by all the participants. It was super clear talk, and I'm really grateful for your efforts with the YouTube show as well. I've often listened and watched, and it helps me summarize the field. So thanks very much. So I will chair this Q&A session now with you, which is essentially, we'll start with some questions that we were given by some of the participants. Hopefully they are here, and it will take it quite relaxed, but yeah, we'll go through some of these. You and I can have a chat about them, and then we'll see where we are with time. So are you okay with this?
Yeah, sounds good.
So the first question, and unfortunately I don't have the information on who asked these questions, but so they are anonymous, unfortunately. The first one is, how do you think senescence is related to organismal aging?
Good question.
Yeah.
How much time do you have? I mean, so besides the sort of standard observation that there does seem to be an accumulation of senescent cells of age, I think that there's obviously more to it than just that, because we also know that senescent cells aren't just seen as a negative thing, but they also have positive roles within the body, right? So they frequently arise during development. And so regards to aging, I think well, so this is one of the key questions is, do senescent cells accumulate because they're not being effectively cleared, or is there just more damage that's triggering them to appear in the first place? And obviously, when you think about what exactly is aging, there are many different ways you can define it. For myself, I think of aging as loss of communication, to use my term I've said a lot today, or in other words, like loss of function. So when the tissue stops behaving how it should. And so senescent cells, because they have entered this unique cell fate, they effectively aren't doing what they're normally doing, and so they're not functioning normally. So in that case, you could say that if there was enough cells functioning abnormally, then that could give rise to tissue dysfunction and therefore be classified as aging. But that's just my personal on-the-spot thoughts.
Yeah. I mean, that's what we're here for. I agree with you. And I think you're right in that it's important to consider that senescence is not all bad. As you're nicely showing, it could be spread around in a good sense. And I think it's a big gap in the field that we cannot effectively distinguish, physiological or pathological senescence at the moment. So following that, would you say that in an aged tissue is where you will find the most senescent cells? Perhaps we can specify in vivo.
Yeah. So I mean, that also comes down to how exactly we go about defining senescence, which, at the moment, I know that there's a lot of work in the space about trying to find the best biomarkers for senescent cells and also the heterogeneity in senescent cells. So I think from my perspective, then how are we distinguishing a senescent cell from an aged cell? And so, yeah, I didn't really have a better answer than that, to be fair.
No, I agree with you, and there are it's frustrating as a researcher that there's no good biomarker for senescence in all contexts. But I suppose that's what our jobs are to explore. So okay, we talked a little bit about senescence in aging. Could you describe the relationship between cellular senescence in cancer?
Yes, I can try. So so I guess there's many aspects to that question. Firstly, there's the observation that senescence itself we can see as a tumor suppressive mechanism. So if you've got some sort of damage, some mutation, so oncogene-induced senescence is something that when there are mutations that are oncogenic, instead of the cell becoming tumorigenic, it enters senescence. And so because it's not replicating, it prevents tumors from forming in the first place. But on the flip side, we have the situation where if you've got a senescent cell present in the tumor microenvironment, it's secreting things like growth factors, inflammatory factors that could therefore be driving further tumor growth. And then I suppose the last flavor of this link between senescence and cancer is that maybe one of the ways in which we can treat cancer is by inducing senescence, which I know that there's research being done on that as well. So I think it's a complicated question.
It sure is, yeah. It is. It's multifaceted for sure. Your final comment about inducing senescence as a cancer therapy leads into my next question as well, which is about the role of senolytics for cancer patients, whether this could be a potential avenue to help with treatments, or maybe we could look at senescence in terms of diagnostics or efficacy of current, chemotherapy treatments or something like this. Could you see a potential role for senolytics in cancer patients?
I mean, I think there's probably better experts in this chat who could give a better answer than I can. But of course, I think my views more generally on senolytics is with the specificity at the moment. So obviously there's some like aging clinical trials being conducted with senolytics, which seem to be promising at the moment, but they're using small molecules, which obviously, whilst they might be eliminating senescent cells could also be having consequences for healthy cells. So I think that there's also exciting research being done on more selective approaches to senolytics. So whether that's through the CAR T cells, so receptor-mediated interactions, or through antibody-based therapeutics. I guess that then comes back to the question about how exactly do you find the right receptor membrane protein to then target the senescent cell. But I think when you have a selective mechanism that's at least antibody-mediated, if you know the targets, if you know the specificities, if you know it's specific, then we know how it's functioning. And therefore, in my head, it feels like a safer therapeutic option.
Agreed. You know, that's great. Thank you. And I know these questions are quite broad, but it's time to discuss them generally anyway. I have maybe a question about the data you showed earlier, if you don't mind. I was wondering if you looked at other markers of senescence in your P53 mutant cell lines, because I know you described the SAS, but I was wondering particularly my favorite protein, which is P21, often is downstream of P53, for example, in senescence. So I was wondering if you looked if there were changes in P21 or other classical senescence markers following a etoposide treatment in your mutants.
Oh yeah, of course. So as I showed with those scatter plots, the dominant negative effect was like the main sort of functionality of having high expression of mutant P53. And I didn't show it, but if you look at the sort of key factor that links all of these genes that are downregulated, it was wild type P53. And so every time I had mutant present, there was always less P21 being expressed. Proteomic level as well. Yeah.
No, it's interesting. I've seen this in histologically in P53 mutant tumors as well. But there is discussion on whether there's non-canonical pathways as well, right, of P21 expression. So
that's right.
Consider. So that will do, I think, for the science questions I have. But we have a couple of questions more about moving to Japan and the first is whether you have any tips for moving abroad and adapting to a new lab culture and culture in general.
So, of course, I have some. Well, I've only done it once, but I have some advice. I think it's important that to try and at least visit the country you're going to before you decide to make such a big decision, and also remember as well that you're moving your whole life, not just moving to a new lab. So for me, stability was something that I made my priority. So making sure I knew I had somewhere to live, knew what exactly forms and everything, health insurance, all that sort of visa side of things that needed to get done. And then also, as I mentioned in the presentation, like having a sort of social support system out here as well was really important to me. And I'm still trying to grow it out here. I mean, so far I feel like it's gone very well. But you do have to be very proactive about it and be that one who always asks first. And then obviously alongside with that, I'm in a country where English isn't widely spoken here. So it has encouraged me, and I'm trying to, in addition to learn about the new science, also learn a language at the same time.
Yeah, no, it sounds like you've done very well considering when you moved 3 months ago. Sorry. I echo a lot of those tips, having moved to Sweden 6 months ago, though I would say the language barrier is much less.
You mean you don't know any Swedish?
I've not learned much Swedish yet, though I've learned a few words here and there, but everyone speaks very good English here, which is very lucky, but not helpful for me learning a new language. So a final question is whether you, on the other side, have it could explain some challenges that you face while applying for roles abroad. And the second bit was what led you to your current position, but I suppose you've discussed that a little bit, having visited Japan before and things like this. But
yeah, exactly. I mean, it's a good question. So actually, one of the biggest challenges I found, I mean, obviously, if you want to apply for a job, interviews are always challenging no matter where you're looking. But one of the things that came up because I was making such a big change was convincing people that I was being serious, that I actually wanted to come to the country. So I guess on paper it was quite surprising that I wanted to leave the UK. And then on the other side from that, I suppose one of my criteria for joining a new lab was to have the ability to learn new techniques. Now, often as a postdoc, you hire postdocs because they come with their techniques that you can then exploit, whereas I wanted to learn something novel. So trying to get a job when I technically don't have the skills, like I need a lot of training to begin with. I guess I can come with my ideas, but I still need a lot of training to get going. So I feel like a student still. But so yeah, so they are probably the two challenges in terms of just general interview process. But otherwise, I think it's just a universal applying for jobs is hard, but it's always also both ways, right? So as much as they're interviewing you, you have to sort of interview them back and work out, is this really the right job and place for me to be to?
Yeah. No, I agree 100%. Yeah, okay, thanks very much, Eleanor. I think with that, looking at the time, we will close the Q&A session.